Okay, I'd like to start by thanking Ali and the KCNT1 Foundation for the invitation to participate on today's research roundtable. So I'll tell you in the next 5 or 6 minutes about two topics. I'll give you a brief overview of our company and technology, and how are we thinking of applying this technology for this grant that we're about to kick off with funding from KCNT1 Epilepsy Foundation and CURE Epilepsy. So a little bit about our company. So we're a preclinical-stage CNS drug discovery company. We're located in Cambridge. We were formerly Q-State Biosciences, and a couple of years ago, there was a change of control, and we became Quiver Biosciences. Our lead assets are two antisense oligonucleotides, one targeting NaV1.7 for neuropathic pain, and the other one targeting UB3A, and this is for Dup15q syndrome. So at the core of the work we do, it's this functional platform that combines three components, right? Human neuronal models. So we work with human stem cell derived neurons. That's my background. I train as a stem cell biologist, learning how to differentiate different flavors of neurons. And then our core technology really relies on our ability to measure with high throughput. With high throughput means the electrophysiological activity of neurons. So we generate large data sets that represent a great input for machine learning approaches to derive insights that can drive both phenotypic discovery and drug discovery and development. So how do we do this high throughput optical physiology measurements? So the technology relies on the expression of two key proteins. One of them is an optogenetic protein called sheriff. So we can deliver constructs into the human stem cell derived neurons expressing this optogenetic protein. And in essence, it acts as a voltage actuator. So it responds to blue light, and in response to that, cells fire action potential. And then the other core component, it's actually the voltage reporter, which is called quasar. So this is an engineered protein that reports on membrane potential through changes in fluorescence emission. So in essence, what we're doing is with special fluorescence microscopes that we built in-house, we can record movies of neurons flashing fluorescence. So technology makes a reality the way that electricity usually gets represented in cartoon as flashes of light. We're converting an electrical signal into an optical one. So in a single movie, we can capture the electrophysiological profile of hundreds of neurons. And then our analytics team has built a custom data analytics pipeline that what it does is deconstruct on all the sources, all the data points in each of the movies. So in each of the movies, we collect data from around anything from 100 to 300 neurons. We get the different electrophysiological trace along the blue stimulus protocol. And then using this information, we can derive a lot of functional properties associated mostly with spike timing and spike shape properties. So at the end of the movie, what you have is anything from 500 to 1,000 different features for each of the neurons that we measure. So our current throughput, it's around anything between 500,000 and 1 million neurons per instrument per day. So we collect large amounts of data. This is a high-level view of some of the data outputs for some CNS disease targets. For a couple of these targets, TSC2 is a tuberous sclerosis gene, and FMR1 is a Fragile X syndrome genes. We have a couple of recent publications that showcase a little bit the utility of the platform in driving phenotype discovery and drug screening. Now, in the context of KCNT1, we have established some neuronal reagents, primarily by using CRISPR-Cas9 tools. So in-house, we have taken a couple of our control iPS cell lines, and we have introduced KCNT1 genetic variants. We mostly focus on two variants, the A294T and the G288S. So doing some preliminary measurements of the functional properties of these neurons, we have identified a few different functional features that are associated to different degree actually to the expression of these two different KCNT1 variants. Here are some of those features we find increase after hyperpolarization, and you can see that the effect is larger in neurons expressing this A934T, which is associated actually with I think more severe clinical manifestation in patients. We also have increased Rio base. So this is after a couple of rounds of measurements. One thing that I won't have to present today is that we have done similar type of work as well in another interesting disease gene called SCN8A, which encodes for NAT16. So what we want to do with this collection of neuronal reagents is now, with funding from the KCNT1 Epilepsy Foundation and CURE Epilepsy, actually explore a little bit more this potential mechanistic linkage between KCNT1 and SCN8A, as Ali was pointing out during the introduction. Here's some of the motivation, right? Both of these genes, KCNT1 and SCN8A, they encode ion channels that are linked with severe childhood epilepsy. For both genes, gain-of-function mutations lead to increased activity of the channel. And then what's interesting here is that increased activity of NAT16 due to gain-of-function mutations could potentially lead to increased activity of KCNT1 in neuronal compartments where both channels are expressed. So this is a potential linkage between these two targets. And there is already some proof of concept, mostly coming from Dr. Miriam Meisler's work at University of Michigan, that shows actually that you can downregulate one target to try to modulate the phenotypes of the other target. So the overall goal of this grant that's sponsored by KCNT1 Epilepsy Foundation, CURE Epilepsy, is to use some of the human neuronal reagents that we have established in combination with our platform to further explore the relationship between these two disease gene targets. And to validate some of that and understand some of that relationship, we're going to use antisense oligonucleotide that we have also designed and synthesized and already tested in some of these neuronal models. And I'll end with this slide to just describe sort of the three modules of work that we're doing as part of this grant. The first one refers to the actual generation of the neuronal reagents. So we have stocks of stem cells that are ready for of different genotypes. So for each of the targets, we're going to use both CRISPR isogenic reagents and also patient-derived reagents, so that we have a large collection of cell lines to ask the question about the functional phenotype associated with each variant. Aim 2 is the actual application of the functional measurements in all of those neuronal genotypes. So we'll do at least a couple of rounds of measurements to define the collection of functional features associated with each target. And Aim 3 then refers to the actual modulation of each of the targets with antisense oligonucleotides. So in the KCNT1 model, we'll test both KCNT1 ASOs that we have already validated molecularly and SCN8A ASOs. And conversely, on the SCN8A as well, we'll test NAT16 ASOs and KCNT1. So with that, I would like to again thank the Foundation and CURE Epilepsy for facilitating the funding of this program that we're about to embark on. And I look forward to in future meetings presenting some of the results of the work as we start generating results.